next generation sequencing methods that we are discussing has revolutionized the biotechnology sector. We have been discussing about the Illumina sequencing method, and this is the workflow of the NGS. The workflow, the first thing is the isolation of the genomic DNA, the fragmentation of genomic DNA and tagging of the DNA with the adapter molecules. So, we continue from there. The first thing is the library preparation. As I said, the genomic DNA is fragmented by using various physical methods. Then the end filling occurs, creating blunt end fragments, and then tagging occurs by attaching the known sequence adapter molecules on either ends of the fragments. These two can be clubbed together in a process which is termed as tagmentation. Now, coming in detail about the cluster generation, the single fragments that have been generated are basically loaded onto what is called as a flow cell. Now, flow cell is a channel for absorbing this mobile DNA fragments, and it is also called as the sequencing vector vessel. So, you can see that this is how the flow cell looks like. It has got these channels which are and where their DNA is basically loaded. Now, an Illumina flow cell is a hollow glass side with one or more channels or lanes coated with oligonucleotides which are complementary to the sequencing adapters, so that the single stranded adapter ligated DNA fragments can attach through hybridization. So, these flow cells contain the adapters. As I said, to the fragments we attach the adapters whose sequences are known. So, the complementary to what adapter we have added to the fragment, we attach it to the flow cells, so that when the single stranded DNA fragments, the fragments of which we want to know the sequence, is run over these flow cells, through these flow cells, lanes and channels, then the hybridization occurs. We provide the conditions so that the adapter molecules which is attached to the oligonucleotide fragments causes the fragment to hybridize to the adapter molecule which is attached to the flow cell. So, this is how basically it looks like. So, here you can see that this has been there are three parts which has been shown. So, as you can see on the channel on the glass slide, there are various red and green adapter molecules that are attached, and a single fragments now come and attach to the adapter molecule. The DNA fragments are hybridized to oligonucleotides on the flow cell. Basically, these covalently fixed complementary strands are synthesized starting from the oligonucleotides, and the original fragments are washed away. So, the first thing that occurs is to make the fragment adhere to the flow cell where the sequencing needs to be occur. Now, you can say, why do not we sequence the fragment that is originally come? Why do we want to create a complementary strand attached to the adapter which is fixed to the slide? So, it is fixed to the slide, that is important. So, we need the strand to be fixed to the slide so that the subsequent procedures, it will not just get washed away. So, the adapter, but we need to synthesize the fragment which we want to sequence. So, we give that fragment, it attaches with the adapter molecule, we synthesize the complementary strand to the strand that we need to sequence, and then we remove the original strand. So, now the strand, the sequence which we, the strand of this which the sequence needs to be known is now fixed onto the slide. So, this becomes very important. So, these are the initial steps. Then subsequently what is done is bridge amplification occurs. It is an important aspect of Illumina sequencing platform. What is this bridge amplification? For bridge amplification, the DNA fragments are bent by hybridizing their second adapter to another oligonucleotide. So, you can see the fragment that is coming, as I said, the adapters are attached to either ends. Now, they can be the same adapters or they can be different adapters. It depends upon us, because adapter sequences we are only designing and putting it onto the oligonucleotide that needs to be sequenced. So, the adapter molecules, there are other adapter molecules also attached to the slide. Now, the top one adapter molecule will bend over and hybridize to the adapter that is attached in its vicinity. So, when it bends and attach, then the complementary strands are synthesized beginning from the second oligonucleotide. So it is very clearly shown here. It starts from the right hand side of the slide, that you can see the bending has occurred. So once the new complementary strand has been synthesized and the original strand has been washed away, then we give the chemical conditions so that the other side adapter also attaches to the slide and creates a kind of a bridge, and then the synthesis will take place. So the synthesis will take place, and then again it will stand, and in this manner this kind of these steps are repeatedly occur, so that at one place you have got multiple copies of that particular fragment. Bridge amplification is used to create small clusters or colonies of identical fragments by repeatedly bending and fragment each fragment such that the second adapter hybridizes to an oligonucleotide in its vicinity, used as a template to create the complementary sequence by extending the second oligonucleotide. So this process repeats itself again and again, so that a cluster of fragments occur, which are identical fragments to the original fragment, the complementary fragment that has been created from the original fragment. So what is the use of this bridge amplification? The purpose of this bridge amplification is that it amplifies the signal that is occurring. Finally, the two strands are separated by denaturation. This is repeated to form a cluster of DNA fragments, of which one of the two orientations is basically finally removed. We just need the single strand which is there. So this kind of a thing occurs so that at each spot identical cluster of fragments is obtained. The purpose of the process is to amplify the signal intensity of the base to meet the signal requirements for sequencing. There has to be a threshold of signal that can be detected. So we start with, only as shown in the figure, we start with one fragment at each spot, but by this bridge amplification we get a cluster of fragments at each spot which is identical to each other. At like 1, 2, 3, 4, these are the 4 spots that are there, and in one there are multiple fragments of that one fragment that has been added on to there. So in that way the entire genome, or whatever we need to, if we are going in for whole genome sequencing, then all the fragments are basically located on this flow cell, and each fragment there are many clusters that have been formed. So this is the second step, which is the clustering. So fragmentation, tagging, clustering, and the last step comes, which is termed as the sequencing. After bridge amplification on a planar flow cell, one of the 4 nucleotides is passed through the flow cell under conditions that favor extension. Now nucleotides are fluorescent terminators, so that only one base is incorporated. So what happens is that to start with only one of the 4 nucleotides is passed through. Now this nucleotide is a terminator carrying a particular fluorophore. What do you mean by now this terminator? As I said, this is a reverse terminator, so let's look at that. We already have seen this Sanger dideoxy ribonucleotide, and this dideoxynucleotide contains H in place of the OH, and then this is nitrogenous base is attached to a di or a fluorophore with a linker molecule. This is how the DDATP or DDGTP looks like. So it can be either, the base can be adenine, thymine or whatever, but that base is basically attached to a particular fluorophore di, and this is a termination group because it has H in place of OH, and therefore it cannot be extended. In this case, this 3' position, there is a reversible group, terminator group that is attached. So OH is not replaced by H, but OH is replaced by OR. And this OR is basically that there is a reversible terminating group that is there. And the linker molecule which is carrying this fluorophore or the di attached to this base is again cleavable. So when the reversible reaction has to take place, then this R will be replaced by the edge, So H, so it will become a normal extensible nucleotide, and this fluorophore will then be removed. So what happens is, when you add a particular nucleotide, then the chains will be terminated at that point and the signal will be detected. But then this R will be replaced by H and the fluorophore will be removed. Therefore, one nucleotide has been incorporated, which is now extensible. So you add the second nucleotide containing a different fluorophore, for example. Again, it will be a reversible terminator group. So now this nucleotide will come and extend wherever the complementarity is there and again terminate the chain extension. Then you reverse the chain termination. You change the condition so that the reversible terminating group is removed, H is attached and the di is removed, again making it a normal nucleotide. Then you again add the third one. So that is how the sequencing is done by synthesis, and as each nucleotide is incorporated, it is read by the machine because it is carrying a particular fluorophore. Then you remove this fluorophore and you make the chain extensible. Then you again add a terminating group, and that is how the series occur. So this is basically what the sequencing, how the sequencing by synthesis occurs in the NGS platform. Each nucleotide has a different fluorescent label so that each can be distinguished by color. Furthermore, the fluorescent terminators are reversible, which means that the blocked 3' end can be regenerated by simple chemical means provided by the flow cell. So, again I explained this, that first of all you add a particular nucleotide carrying a fluorophore. It is a terminating group, just like in Sanger sequencing. It will terminate the chain extension, but then this is carrying a reversible chain termination group and not that the OH is replaced by H. So based upon the conditions that are in the flow cell, this reversible terminator group will be removed, which will cause the cleavage of the fluorophore molecule, and the nucleotide will become just like a normal nucleotide because the reversible terminator group will be replaced by H. It can now be extended. So in the second cycle, you add another fluorophore containing nucleotide, it will attach. So this will read only those fragments where the complementarity is there, but you can go ahead and keep on doing this. So each cycle involves adding polymerase and DNTPs as fluorescently labeled terminators under conditions that favor extension. Then you wash the flow cell, you image the fluorescence, you cleave the fluorescent terminator and then wash the flow cell and then again repeat. So at every step, every point after the cluster has been created, repeatedly you keep adding the polymerase and DNTPs as fluorescently labeled terminators. So simultaneously you can add all the 4 as well, containing different fluorescences. So they will bind at different places based upon the complementarity, that what is the nucleotide that is coming next. Then you wash the flow cell to remove any unbound fluorescence that is there. You image the fluorescence and then you cleave the fluorescence and again add the polymerases and the nucleotide. So this way the image is produced. So for example, this is showing one of the steps when you add all the 4 nucleotides, that on certain fragments, whatever the complementarity is there, it will contain, that fluorophore will be added and then the image will be there. So you can see there is an image of a green, a black, a yellow and a yellow, blue and a red one. So this way, this is one nucleotide that has been read. Then again you remove all the fluorophores, you add the DNTPs, then the next nucleotide will be read. Again you remove it, again you extend it and then the next nucleotide will be read. So in this way, all the nucleotides will be read and the image will then be translated into a text file. And this text file will then contain the sequence of the different reads. So now these are basically termed as the shorter read. These are short read molecules. So these fragments will then be read, and as I said, you can see that the signal has been intensified because of the clusteration that has occurred, and then each fragment can be present multiple times and therefore multiple reads. This is called as deep sequencing, that a particular fragment you keep sequencing again and again, because these are randomly distributed fragments, so they can be read from different places. The same fragment can be read. So this is how the imaging occurs, and then it is basically changed into a text file that is there. So sequencing reagents, including fluorescently labeled nucleotides, are added and the base is incorporated. The flow cell is imaged and the extension from each nucleocluster is basically recorded. Emission from each cluster is recorded. The emission wavelength are then filtered and basically read into or transported into a different kind of signaling text reading, and then it is basically the cycle is repeated, and thus it creates a read length of n number of bases. So this is basically n times it is repeated, so different read length of n bases are basically incorporated. Then when you get multiple reads, then these reads are aligned together and they are also aligned to a reference genome. So again, when we talk about how these are, when we talk about the whole genome sequencing, then how these reads are assembled to form a complete genome, that is a different aspect, and we will discuss when we start with the bioinformatics series, where we will discuss that whatever data that we have generated, how this sequence data can be assembled or validated and be give rise to the entire complete genome sequences that we can get. So in here basically all the reads you can see they are aligned together, and they can also be aligned based upon a reference genome. So there are two kind of aligning that occurs. One is based off, so there are algorithms, these are all machines that they do. So there are algorithms that will basically align them. There are aligner softwares, so they will align all the reads, so they will filter the reads, they quality control the reads, then they will align the reads, and then they will assemble them, because then they have to join together all these reads to give rise to a particular genome. And this assembly can be done by de novo method or by using a reference genome. So all this we will discuss in our bioinformatics series, that how to analyze this data that we have obtained. So for now, this is basically how the alignment and data analysis occur. So this is basically the Illumina sequencing workflow that has occurred. So we can know the sequence of each fragment. Now there is another term which is used, which is called as a paired end sequencing. So paired end sequences again gives an advantage, as it allows to sequence both the ends of a fragment and generate high quality alignable sequence data. PE sequencing facilitates detection of genomic rearrangements and repetitive sequence elements, as well as gene fusion and novel transcripts. Now it is a very important aspect that when we are sequencing such shorter reads, it becomes very difficult to assemble the shorter reads, because there will be many regions which are present in duplicates. There are many repetitive sequences which are present in a genome. So for that, this paired end sequencing is very handy, because each fragment is sequenced from both the ends. So the alignment of these repetitive sequence becomes easier. And again, various gene fusions or novel transcripts can be detected. So this is what the paired end looks like. So the paired end sequencing, you use the adapters, which are basically, you use the sequencing from both the ends. And when you are aligning, as you can see, if there is a repetitive sequence as well, then because of the sequencing from both the ends, the fragments can be aligned in a better way, so that repetitive sequences can be resolved. So many a times on these platforms, paired end sequencing is done, which gives a clearer and a better picture of the sequencing methods. Now let's compare the Sanger sequencing versus next generation sequencing. What are the similarities to start with? Both contain sequencing by synthesis. Both are basically using the terminator sequences. The critical difference between Sanger sequencing and NGS is the sequencing volume. While the Sanger method only sequences a single DNA fragment at a time, NGS is a massively parallel sequencing millions of fragments simultaneously per run. The process translates into sequencing hundreds to thousands of genes at one time. This is the major critical difference between the Sanger sequencing method. The benefits of Sanger sequencing method is that when you have a single or less number of samples, or the Sanger sequencing comes very handy, because there it is, the cost is comparatively lesser and all those kind of thing, but it is usually used when you have only a single or very less number of samples that needs to be sequenced. However, the targeted NGS, as we said, it has got higher sequencing power, it has got higher discovery power, it has got higher, it is a very deep sequencing, it is higher reproducibility, it has got a high throughput sequencing that is required, the sequencing very low amount of DNA or RNA. So we have already discussed the advantages or the benefits of next generation sequencing which is there. There are some challenges, however. For example, in Sanger sequencing, the low sensitivity is a major challenge. The resolution is not that high. The low discovery power, because it cannot, it is not a highly, it cannot sequence at a higher resolution. Therefore, the discovery of new variants becomes limited. Again, it is not very effective if you have very large number of samples which are there. It is not advisable to use Sanger sequencing method. And again, reproducibility, reusability. However, I should mention that Sanger sequencing is still used when you have got one or two samples. You just have to sequence a particular plasmid DNA, particular insert in a plasmid DNA. Then Sanger sequencing comes very, very handy. The targeted NGS, the major challenge is the basically the cost effectiveness for sequencing. Although the very low cost, it has become very cost effective as newer and newer platforms are basically occurring. Then initially it was very time consuming, but again, now it has become much more rapid, the new next generation sequencing methods. So these two are continually being researched and developed, so that to have more advanced NGS platforms where the cost is also lowered down and the time is also cut, and it can happen very rapidly. So this again tells you like how do you choose between the Sanger sequencing or the next generation sequencing method. So if you are looking for variants, and that too in a very less number of samples, so you can see vertically it becomes low to high, the resolution, and on the horizontal platform it's basically the variant screening to the discovery power. So when you start with, you can basically, if you just have to talk about the less number of samples are there and you want to screen even for variants, you generally use the quantitative RT-PCR or you use the Sanger sequencing method along with that. But if the number of samples is high, you go for the next generation sequencing method. Now, next generation sequencing method again depends upon like how much amount of DNA you want to be produced. As you go towards the right, we are dealing with the not just the amplicon sequencing, but we are dealing with the enrichment sequencing. So enrichment sequencing is when you enrich a particular kind of fragments that you want to sequence, or any functionally related genes that you want to sequence, you enrich those clusters and then you sequence them. Or you go in for, as I said, the amplicon sequencing, or further you go in for the whole genome sequencing. So for the whole genome sequencing, you go for the NGS platforms. Whenever the sample number, even if it is low or high, there is more chances of discovery of the new variants or the discovery of various parts of the whole genome sequencing which is there. So this is how you choose between the Sanger sequencing or the next generation sequencing, and what type of next generation sequencing is required. So type basically means that amplicon sequencing, that you generally want to amplify a particular sequence. For example, if you want to just know the microbial diversity, you can in a bacterial diversity, you can just amplify the 16S rRNA gene. You can use universal primers, you can just amplify 16S rRNA gene and you can sequence them, so that you can know the particular diversity in a molecule, in a sample. If you want to go in for a functional aspect, then related to that functional aspect, you can enrich the clusters that you are using. So that is basically the enrichment culturing. And then, if you want to just know the entire genome, you can use the whole genome sequencing, which is there. So again, for discovery or for variant screening, you can choose between either the Sanger sequencing or the whole genome sequencing methods, which are basically there. So these different sequencing methods can be employed. Now let's look at the last, the applications of these NGS. Now NGS allows the research labs to rapidly sequence the whole genomes. Now as I said, the first human genome took 13 years to be sequenced and costed around such a high rate, so many dollars were costed. Nowadays we can get our own genome sequence within a week and costing very less, not much. So it depends. Again, if you just need diversity, you can probably get it for 10 to 11,000 per sample. If you need more, you can cost up to 30,000. So for a whole genome sequence, my own genome I can sequence within a week for costing as less as probably 30 to 35,000 rupees. So it is a very rapidly sequencing whole genome method which is there. As I said, it deeply sequence target region. So deeply means that multiple times it sequence. So therefore there is a high throughput sequencing that is occurring. So it creates, it generates a lot of authenticity in the sequence that we are getting. It utilizes the RNA sequencing, which is the RNA-seq method, to discover the novel RNA variants and splice sites or quantify mRNAs for gene expression analysis. So rather than using DNA as a source, RNA can be used, and RNA sequencing can be done, which will basically talks about the gene expression analysis, which is there. It can analyze the epigenetic factors such as genome-wide DNA methylation and DNA protein interactions. So this is again, NGS platforms are used to understand these epigenetic factors. It can sequence cancer samples to study rare somatic variants, tumor subclones and much more. It can be used to study the human microbiome, and also it can identify novel pathogens. So these are some of the applications of NGS platforms, which, as I said, has basically revolutionized the biotechnology sector. It has revolutionized the way we understand the DNA, the RNA and the functioning of the cells, and it is, the advancements and the development of newer and newer techniques is making this this technology much more advanced and for a better commercial uses. So that's all for today. Thank you.